and etc. And today we're joined by Ville. I think we're going to give the same introductions of the people that are up here. So Jean-Paul, you want to introduce yourself? Jean-Paul. Maybe he's not there. Serena, you want to introduce yourself?
Sure. I am the Research Development Director at Taylor. We're a service center at Baylor College of Medicine. We provide phages for compassionate use clinical cases.
And we're also joined by Austin.
Yes, hi, I'm Austin, the Director of Operations for Taylor here at Baylor College of Medicine. Manage everything that is needed to bring phage cocktails to patients on a compassionate use basis.
And also joined by Justin. Hola, Justin.
Hi everyone. Yes, I'm Justin. I'm also with Taylor. I am the lead data scientist for Taylor. I work with the genomics and basically anything with analytics.
And I'm Adriana. I recently defended my PhD, my dissertation in the Center for Phage Technology, and right now I am in limbo, which is great when you have to wait for papers, but it's great. We're also joined by Jean-Paul, who might not be able to unmute himself currently. He does phage therapy in the Queen Astrid Hospital in Belgium. He's already talked about the Belgium Magistral Phage Corporation and their idea of phage therapy. And today we're joined by Ville. Do you want to introduce yourself, Ville? You have a pretty cool CV, by the way.
Yes, thank you. So my name is Ville, and I work as a lecturer in University of York, and I'm working on quite a variety of topics, but I've been working maybe longest with phages, and I'm kind of basic biology, but I'm ecologist and evolutionary biologist by heart, and that's what I do most of the time.
See, sorry, Ville. I didn't know that your name was pronounced Ville. I'm so sorry. He's also a cool musician, by the way. He's cooler than at least me up here. So can we start, Serena?
Yes, we're recording.
Good. So that everybody knows, we're recording the conversation, and we're going to be making it available later in our Facebook page.
And if you have questions and you don't want to talk, you can press that little arrow that's in the corner of your screen, and you can just text your question, and we can tell everybody what that question is, and you don't have to talk.
Also, we welcome everybody and questions up here. So this whole thing started because we saw a paper from you published about training phages, and that's a cool idea of phages, and especially from an evolutionary point of view, it's very, very cool. Ooh, Jean-Paul was there. Hola, Jean-Paul. Jean-Paul. But whatever. So yeah, we saw a paper from you about. Go ahead.
I'm sorry. I had to invite myself as a speaker. Apparently, it's a bit strange, but so now it works.
Well, sorry.
No, it's a bit strange. I could invite myself as a speaker, but I couldn't speak.
Oh well, the things about doing this live. There you go. So I was talking about the phage paper where you actually talk about training phages, but there's a lot more work that comes before that where you look at dynamics in populations in the rhizosphere or rhizosphere, however you pronounce that, sorry, and evolutions of populations of bacteria and fungi, and then I don't know if there is any phage work involved in there, but it seems that you've done some phage work in combination for like wilt tomato disease and stuff like that. So you want to tell us a little bit about your evolutionary work before we go into training phages?
Well, I mean, like I can give a kind of brief overview. So I kind of started as a kind of pure like experimental evolution, do some evolution in test tubes kind of approach, which is of course quite simplified environments. And then and then I started this with sort of clinical context, and with Pseudomonas aeruginosa and aeruginosa-specific phages. And it has kind of evolved through the years to, I've always been fascinated by communities and how complex and difficult they are to understand. And then just kind of out of like, what happens in the test tube in the lab, what is the relevance in natural systems. So it's kind of developed towards more natural communities and on rhizosphere. And there we've been focusing on plant pathogenic bacteria, especially one called Ralstonia solum serum, which is quite interesting pathogenic, can infect lots of different crops, different plant hosts, and it's genetically quite diverse, and you can find it almost everywhere. And then of course there's a bit of different problems there, but you can apply phage therapy in sort of in vivo context using plant microcosms and greenhouse experiments. So it kind of offers you more sort of, I would say, maybe a bit more direct route to actually, from the blue sky ecology, evolution, biology, to actually try to translate phage therapy into agricultural practice.
That's super cool because you can also determine, not just in the lab, but also outside of the lab, what is the fitness cost of becoming resistant, phage resistant. There's a couple of you have where you participated in where you talk about resistant evolutions and how that affects communities in general, bacterial communities in general.
Yeah, so I would say what we try to do a bit is also to look sort of safety. So just kind of working with some companies. Of course, one way to establish the phage therapy, you need to be sure that phages are targeting your focal pathogen and you don't have any sort of collateral damage to the surrounding microbiota. And I think I'm not sure in agriculture, but it seems that there's no really clear guidelines how to verify this. And of course, it's so context dependent. The microbial communities are quite different with different crops and different environments, and also they vary similarly between different humans and different hosts. So we can do a bit of work, try to see what happens when we apply phages in sort of communities, model communities in the lab and the rhizosphere, and then do they kind of exert direct effect on the other bacteria, just directly testing if they can infect multiple different other taxa you can isolate from the soil, and then also looking sort of indirect effects. So you kind of play on sort of competitive release when you have phages that are targeting the focal pathogen that can actually be quite dominant member in the community. You're kind of releasing niche space for other bacteria to take up. So you're actually playing on this sort of indirect effect on the community assembly and so forth. So actually removing one species is also having some effects on the community and community functioning.
So this means that on the long run, and I'm going to put it a bit in context in terms of phage therapy here and in humans, is that removing species from the gut microbiome or the lung microbiome would also have an impact on the rest of the species within that microbiome that we have not accounted for. Am I correct?
Yeah, probably you might have some indirect effects. It probably depends on the sort of how dominant your target bacteria is, but it will definitely, removing that one species will open up space for other species to enumerate and occupy in. And then I'm kind of interested in, from the human perspective, we've been working on the cystic fibrosis sort of model lung community, which is often quite diverse as well, and you have lots of sort of pathogen-associated species that are not necessarily causing infections, but they tend to be associated with Pseudomonas, for example. And then I'm kind of interested in as well, like what is the interaction of these species with the pathogens? So should we only target the focal pathogen, or should we also target the species that are associated with the pathogen? So we have some evidence, for example, that some species might help other species by breaking down beta-lactam antibodies, and sort of because that kind of creates sort of exposure protection near vicinity of the antibiotic degraders. So it might be actually the presence of other species might be helping out the focal pathogen, and actually by killing its sort of beneficial friends, you might be able to help to suppress the pathogen as well. So this is all this sort of ecological feedbacks, and I'm thinking the kind of community, thinking the phage therapy at the community level.
This is super cool. And so now, in terms of phage and phage resistance, do you see any dynamics changing with the appearance of phage resistance in those communities?
Yeah, so we do see resistance evolving, and we, for example, in the soil, we see resistance popping up. Of course, sometimes it's hard to differentiate this from the de novo evolution or sort of community sorting and ecological sorting. So you might have lots of sort of standing variation and presence in complex communities. And it might be that when you apply phage, you're just kind of selective for the already resistant spontaneous mutants that are present in the community. And I guess this is the case in clinical case as well. You do have probably resistance just evolving randomly because it's basically correlating positively with some other traits. So I think for this sort of phage training perspective, so I think we've done some work with clinical strains and some model strains, and it seems to be quite clear that it's hard to evolve increased infectivity against a wider range of pathogenic strains necessarily. They don't all respond to phage the similar way, and then there seems to be also limitations in terms of how infective they are across different genotypes. So it might be that even if you can evolve phage infective to one sort of genotype, it doesn't increase infectivity when challenged or exposed to another bacterial genotype.
Yeah, so you went directly to phage, to the training. And so we wanted to know, so how did you, we know that based on our previous conversation and previous week, there's like various techniques to train phage. How did you train the phage that you evaluated in their work?
So I mean, like the basic principle we've been using is like that simple. You just culture the phages repeatedly against the same host. We've been doing that with several different bacteria and phages, different species. And then basically the idea is that we just keep the phage evolving. So we isolate the phage and re-expose that to the sort of ancestral bacteria repeatedly. Of course, you could do this much more complicated ways. And then I'm kind of trying to do this against multiple different bacteria at the same time at the moment, and then try to see if we can at the same time increase the infectivity and then host range at the same time, or sort of select for infectivity against multiple different hosts in the same sort of mix. And then, but like the basic principle is just like you just keep culturing your phages and wait up for the mutations to pop up and then selection to do its job.
And what is the effect of those phages, those evolved phages in the original bacterial strains?
Yeah, so we haven't done much of the sequencing of the phage at the moment. We don't know how it is adapting. In some respect, it's also, we could do the sequencing, but then we probably get some hypothetical proteins, and the phages we're working is they're not necessarily that well characterized. Most of the phages that's just isolated from the wild. So kind of compared to well-established study systems, even though you can sequence and you can find some differences, it might be a lot of work still to figure out what are these mutations doing. So we're kind of working on that towards that mechanistic understanding with Pseudomonas and now more with Ralstonia. But it's tricky.
And in terms of MOI, do the adapted phages then infect better the original strain? Do they infect it worse?
So what we got, basically we tested, for example, binding and sort of efficiency of plating, and it seems to be that we've evolved two different types of phages. Ones that use LPS as a receptor, and the other one that uses type 4 pilus. And we can evolve both of them more effective. And when we apply them as a combination, we also get improved pathogen suppression. So just in terms of bacterial density reduction. And then what we found out that we don't see any sort of increased binding efficiency. Instead, we just see higher phage replication per bacterial cell. So it seems to be that's sort of the mechanism, how they evolve. What are the genes involved? I have at the moment no idea.
This is very interesting. And in these novel phages, can these phages bind more than one receptor, or these evolved phages, can they bind more than one receptor, or do they lose specificity? Like if you have phages initially binding to ELF and then switch binding or binding more tightly, or I don't know, right? Because binding, you just said binding didn't change, it just replication. So
so I mean like we tend to get with Pseudomonas using Pseudomonas against Pseudomonas PO1, so sort of your lab rat. We tend to see this sort of arms race dynamics or sort of escalation of infectivity. So they become better suppressing the ancestral pathogen, but yeah, we see some effect on some positive effect in terms of inhibition with some clinical CF isolates as well, but it's not that great. And you know, yeah, so there might be some costs involved from the phage perspective, but you know, we don't know the mechanisms in terms of how many receptors they're binding. Quite often we also see lots of sort of weird stuff with the environmental phages, sort of like they can form plaques on agar overlays, but you don't see much inhibition in liquid media, in rich media, and it's probably because of some of the receptors are not expressed in your standard lab conditions. It could be also temperature or density dependent, and you know, you get all this sort of natural variation, all sort of like unpredictability in all these interactions when you move beyond your sort of lab species, which is kind of interesting, but it's also taking much more time to figure out what's going on, sort of more mechanistic level.
This is super cool.
I had
go ahead.
A question. Have you tried combining the phage training in more complex communities and then within the environment as well?
So that's what we're planning to do now. So we've been, this is sort of from the rhizosphere system. So we've been evolving phages to become more effective against Ralstonia solon serum, and this is kind of nice. Like we got these phages and the bacteria, they're from environmental reservoirs. We just isolated them, and we've sequenced the bacteria. We'll be sequenced the phage, and we'll be then testing the efficiency of these more sort of trained phages in natural communities. So we plan to run greenhouse experiments to test if they are better than their ancestral counterparts. And then, yeah, so I think that's the kind of benefit of using sort of plant systems or this sort of more controllable. Like, I mean, like the theory and the ideas are the same as in sort of human treatments, but it kind of allows you to test these effects in more natural conditions and see if they actually hold. Maybe we see more costs in terms of phage becoming and evolving more effective, which we're missing in the lab. But then in the soil, we might not see differences because there's some aspects of phage fitness we don't capture in sort of too benign environments.
Did you evaluate bacterial fitness after becoming phage resistant?
Yeah, so we looked at this with Ralstonia and then Pseudomonas as well. And then this, quite often you see fitness differences. You don't necessarily see those in your LP cultures, but with Ralstonia we see really big changes, and then And with Ralstonia, we see, for example, pilus targeting phages. We get mutation in pilus, but often we also get mutations in sort of quorum sensing receptor genes. So quorum sensing system is mutated, and then this leads to sort of quite big cost in terms of basically blocking the virulence. And then these strains become completely avirulent, so you can take the strains and then you can take a needle and infect them inside the plant xylem, but they still are not able to kill the plant. So I think these sort of costs are actually also one sort of hidden benefit of phage therapy. So you can think about sort of steering the phage fitness towards less virulent direction. So the resistance is not necessarily bad if it actually causes high costs and trade-offs with the virulence.
Have seen the trade-offs the way where phage resistance leads to better growth or more virulence of the bacteria? Has that been described?
So we've done some experiments in the lab in the rich media with Pseudomonas, and we found sort of tunnel stress responses where the bacteria evolve resistant to the phage, and they also evolve resistant to the antibiotics. And then mechanistically, this is sometimes linked with the evolution of mucoid phenotypes. So it seems to be the same mechanism, forming basically exopolymers, basically protects the cells from antibiotics, probably reducing the diffusion, and probably also from the phage by blocking potential receptors. We haven't really verified mechanistically what's going on, but that's like one potential hypothesis and explanation. But sometimes we do see this sort of positive correlation against multiple stresses. And then in the other cases, you have increased phage resistance leads to increased susceptibility to antibiotics. And what we tend to see is when we apply LPS binding phages, we do see increased susceptibility to colistin, which is acting on the LPS as well. So I think, this comes to the sort of phage antibiotic synergies, which people have been studying quite a lot. And then we do see all sort of patterns there, positive correlations and then negative correlations. And then probably in this case, it really comes down to the exact mechanisms, what the outcome will be.
This is super cool. I have one last question. Well, not last question, but please, people, feel free to raise your hand and come up and ask questions. There you go. Good, ears up. Josh is also in here, so you can also come up. What about stability of the evolved phages that you have after doing the training? Are they stable? Are they more stable? Because one would think that nature of these phages can actually, so the scope of their evolution can actually range through all of these possible phenotypes or possible binding mechanisms to the bacteria, but then somehow these phages that we have isolated are being selected for in the environment. So have you looked at stability of the phages? Maybe those phages are more or less stable than the ones that you found initially in the environment?
Yeah, we haven't looked at it, but that's a good point. And we've been trying to, when we kind of trying to screen for potentially effective phages, we do kind of look sort of evolutionary things like how difficult or easy is it for the bacteria to evolve resistant to this specific phage. You can use the fluctuation tests, and you can also. We're also looking sort of environmental parameters. Some temperature is one we're kind of interested in. We want to have phages that can work against bacteria at different temperatures. And then with Ralstonia, you tend to find sort of when you increase the temperature, you get less effect of the phage, even though the phage is not necessarily that heavily affected. Phage can survive the high temperatures, but there's something going on with the gene expression and the interaction that leads to the reduced inhibition by the phage. And I think it's absolutely true. There might be less stability, but of course then if the stability is a problem, and then if you're evolving your phages to use in soil or or in a clinical environment, I think the key thing is to try to emulate the sort of within host environment or associated host environments, and you can then do the phage training in sort of environments where it's supposed to be acting in the therapeutic environment.
That's super cool. Thank you for all of your answers. And Gedir has a question?
Actually, I was going to ask about the temperature used in these experiments. If they tested different temperatures, if you tested different temperatures or only the usual one for the growth of the selected bacteria.
Yeah. Go on.
Okay, so because usually phages are isolated from like wastewater, temperature there is like 20, and the bacteria grow at 37, for example. So have you?
Yeah, it is interesting. So, for example, with the sort of the plant bacteria we're working on, the phages come from the UK river network, and it's not too warm. Maybe periodically the water can get warm for a short period of time in the summer, but basically most of the year phages are in quite cold water, but still they're effective up to 35 degrees, which is sort of surprising because you would expect like why would they kind of keep the stability, but maybe it comes as it might be that there's some sort of, if they are well adapted to the cold temperature, they're kind of well adapted in general to environmental stresses. So yeah, but you're absolutely right. We've used phages from wastewater for the Pseudomonas, and they tend to work quite well at 37 as well. So we haven't got any problems there. But yeah, I know that some of the phages are not good at high temperature and might die out.
So training the phages didn't improve their ability to infect at the temperatures, like physiological temperatures, for example.
No, they've been okay. So we've used temperature haven't had effect in our phage training. In our experiment we've done 37 or lower. So it has been surprisingly consistent.
Thank you.
Josh, welcome up. Nice to see you again.
Hello. Thanks for having me again.
You have comments, questions?
Not yet. I think I'm just learning from Bill. Yeah. Thank you.
James, nice to see you.
How's it going? Can you guys hear me?
Pretty good. How are you?
I'm great.
Yes.
Hey, Billy. How you doing?
Good. How are you, James?
Yeah, good. I haven't seen you in a while.
Yeah, it's been a while.
Right. I guess the question I had was on similar lines to temperature. Have you guys thought about oxygen concentration? I think Rostonia can grow anaerobic on nitrate, right?
Yeah.
So I'm playing around with Pseudomonas and oxygen differential, like CF lung, low oxygen conditions, all that sort of stuff.
Yeah.
I'm finding quite a difference in what efficiency of placement and stuff on the phages.
Yeah.
I was wondering if you guys haven't had any chance of the same thing.
Yeah, we haven't got I had one PhD student, we were looking different sort of environmental factors, and then we were playing around with the idea of using the oxygen, but then kind of found it, it's quite tricky to manipulate. You probably come across as well. And then we went just sort of viscosity manipulation in our setting. But I think that's really important work. So what we kind of changed with our sort of Pseudomonas CF lung model is that we use synthetic cystic fibrous media in all of the experiments, just at least try to replicate sort of nutritional conditions which should have effect on sort of mutation rate, potentially cost of resistance and so on. And then, it's still a very, very model environment, but I think, things probably happen very differently if you change the oxygen concentration, and then that will definitely have effect on the sort of ecology and evolutionary interactions.
Yeah, so the main takeaway from what I've seen so far is it seems to be the phage genome size seems to determine how well it'll work at low oxygen conditions.
So higher the genome size, whether they are adapted to different conditions or.
So that was the alternative. My first thing was like, a smaller genome is more likely to get across the membrane. It's an energy-dependent process, right? It requires more force. So anaerobic growth, 2 ATP compared to whatever it is for aerobic growth. I was thinking, it's going to a smaller genome will have an easier time to get across. But no, it's the accessory metabolite genome seems to be controlling anaerobic growth better once it gets inside.
Yeah, that's really cool result. And I mean, like, they have to use some. Why would they have that sort of DNA unless it's beneficial in some conditions? So
they got a massive genome.
Yeah, exactly. Yeah.
I've also had good progress using Freya's pig lung model. So if you guys, we've been using SCFM2 as well, but I've had some really good ones. When we can get the pigs, had some really good stuff working with that model. The phage will, after put the bacteria in, then give it for 2 days to actually initiate a proper infection, and then add phage later. We're able to drop a couple logs still, and better than any topramycin or Cipro, any antibiotic. So that model works. If I'd suggest if you're interested in more CS stuff, maybe use that model as well if you can get hold of the lungs.
That's such a cool model, and we've been discussing with Freya for a long time to try to kind of get it up and running as well. I just haven't had time. But I think that's really cool, and probably the surface attachment and the biofilm formation and clearing of that is the kind of most sort of real you can get. This sort of kind of trade off with the lab systems and doing something with mice.
Yeah. Oh, I mean, I'm still enjoying your work, Karen. And nice use of the word phage steering. Thanks.
Yeah.
Appreciate it.
Cheers.
No worries. Bye.
Bye.
I have a question, if I could.
Of course, go ahead, Austin.
Yeah. So, really, I grew up on a potato farm, and Rostonia causes brown rot, which can be a huge issue. So I'm sort of interested in the application of phage for this purpose. I was wondering, do you know if you add Rostonia phage to the soil or plow it into the soil, will the phage end up in the tubers and stems of these plants?
Yeah, so we don't know that yet, but how we've been applying is just along with the irrigation water. So that's how we can add in the phage. You can also infect the phage into the stem if you like, but we've been just okay, what if we add it in the soil? Does it have an effect? And it's probably as uncontrolled and an easiest way you could imagine. And in some cases it does work quite well. And then we're kind of trying to build a bit on this sort of translational potential, like adding the phage once or adding the phage continuously, and try to see if we can get it work. So kind of currently running some greenhouse experiments using tomato, and doing these experiments. And we've done some bit of field work in China as well. And they trialed different phage combinations. So we try to increase them, because it's sort of wild environments, we don't really know what sort of bacteria we have there, what sort of genotypes. So just using a maximum 4 phage combination, try to see if we can have any effect. But just the scale of applying phage and manufacturing those amount of phage, to be honest, I'm not sure if it's feasible in the end of the day. But we do see some positive effects, so I'm hopeful.
Yeah, that is great. Thank you. And do you know? Have you found out or tested whether or how different types of soils can affect the activity of the phage and phage cocktail you are using?
Yeah, so there's a long-term experiment which started just about 1.5 years ago in China. With my colleague Wei Xiong has established this sort of soil microcosm, mesocosm experiment. So instead of using, I don't know if you come across this sort of mesocosm warming experiments where people use these sort of small ponds and then expose them to elevated temperature. So basically using, just like we have containers of soil and different soil types that we culture in tomato there, I will look in sort of rhizosphere microbiome assembly and then also do experiments by applying phages. It's going to be a huge long-term experiment. So testing the soil type, but other than that, we haven't really tested that specifically. But we have sampled natural soils quite a lot and done recently some metagenomic sequencing as well, try to establish like what is the sort of the rhizosphere phage community like. And we have kind of in natural environments without any manipulation, we've been able to kind of establish that healthy plants, they tend to have higher abundance of Ralstonia phages. And then we also see interesting sort of temporal fluctuation between different Ralstonia phage types. So some of them are at higher abundances at early plant development, and then some other will become more dominant later on. So it seems to be that there's sort of phage community assembly during the plant growth cycle. And interestingly enough, there's loads of other phages as well that are also associated with the sort of healthy or diseased plant outcome. And then they're affecting other bacteria that might have beneficial or antagonistic effects by interaction with the pathogen they're interested in. So it seems to be quite complicated, but at the same time quite predictable in some respect as well, which is I think that's the main thing. If we see something that is predictable, we can work on that, even though we wouldn't understand all the details.
That's great. Thank you.
Does anybody else in the audience have questions?
There is 2 anonymous questions. So, first question, have you tried to use phage training to train temperate phages to be more lytic, or to be lytic?
We haven't done that. We have some temperate phages for Pure 1 in the lab as well, and I haven't thought about that. I'm not 100% sure how to do that, how to select for that. But maybe, yeah, I mean, like this really cool paper I read yesterday, one Cooper's lab, where they're going to show that when you combine different pseudomonas isolates in the same sort of wood model, and they start competing, the phages start switching around, and then they're actually driving some of the genetic changes and phenotypic changes. So it's not driven by SNPs, by phages jumping across between different competing genotypes. So I think, probably the temperate phages and then their role is a bit of underlooked in a way. And I think, like in general, like mobile genetic elements and how lytic phages might induce that. It will be something interesting to look at more detail in the future.
Okay, and then the second question, is it possible to evolve genes that are more highly conserved, like integrases or repressors?
To be honest, I have no idea. It's a good question, but I can't really answer that.
Anybody else have more questions? I'm going to ask a practical question. From the evolutionary biology perspective, what is better? To treat bacteria with a cocktail of phages from the start, or to treat them sequentially with a cocktail of phages? That's part kind of 1.5, because the first part of the question would be, how predictable are these evolutions in phage resistance / phages, phage affinity? Are they predictable? Can we predict that?
I guess like both approaches could work. Probably depends on the sort of type of host, like the host parasite coevolution and the dynamics. So I mean, like if you have sort of fluctuating dynamics, just using 2 phages sequentially might be able to kind of keep the pathogen still, so it can't really, and in this example I'm just like thinking evolving resistant against one phage will make you susceptible to the other one. So, you wouldn't necessarily need all the phages at the same time. And then if you don't apply them at the same time, you don't exert selection for some other types of mutations that would actually make the bacteria resistant to both types of phages. I guess like with the phage combinations, also if you don't apply the phages for too long a time, maybe these are quite effective, because it really requires some super mutant with multiple mutations against different bacteriophages binding different receptors. So likelihood of that happening would be quite low. But you probably these sort of therapies would need to be, fitted with the sort of system you're working with and sort of treatment length. So, I think that the problem with resistant evolution, probably it's not something that's necessarily going to be a problem during the treatment. If you think about the phage treatment would be sort of 5 days or 8 days, and that's it. But over the long term, probably you might have selection for sort of pathogenic strains that are also able to resist phages, and then they might become more common when you apply phage therapy as well.
Thank you so much. Are there any more questions? Any more anonymous questions? Jean-Paul?
Hi, Ville. So I have a question about this phage resistance eliciting less virulence and more susceptibility to antibiotics. Do you think this is also possible in vivo? Because a lot of those papers, they take virulent and antibiotic-resistant strains from patients, and then they evolve phage-resistant clones in vitro without any pressure of antibiotics or immune systems. Do you think that it would also work in vivo?
Yeah, it would be really interesting, depending on of course, how you treat, what is sort of the location in the body you're treating. But you might be exposed to sort of antimicrobes produced by the immune system as well. And in reality, probably there's some sort of selection from different fronts with the bacteria as well. Yeah, but I think the kind of combination therapies, I think that's our kind of best bet. I think with the sort of evolving sort of against sort of kind of keeping our treatments to work long enough and prevent sort of resistant evolution. But I think in reality, bacteria will probably more resistance to whatever we kind of throw at them. And in case of sort of cystic fibrosis and weakened immunity, I think there's probably a limit how far we can stretch any sort of treatments. But yeah, I think we need honestly more data, just looking the sort of what happens, phage and bacteria evolution within host type of environments. So I think, yeah, I guess it's I'm not really sure.
Well, we see, of course, a lot of synergy between phages and antibiotics, but we don't think it's necessarily linked to resistance. They just work both together better than alone. So I'm not sure that an antibiotic works better because there is phage resistance, because we see this effect from the beginning also.
Yeah, in vivo, yeah. It could be just correlation. I think with the probably when we do experiments in the lab, we can start everything in a control setting. We have sort of isogenic populations, and we can drive it in different directions and then explore how it affects fitness when we change the environment. I guess with the in vivo, we don't know what we're working with to begin with. So probably inherently all the outcomes will be much more complicated and unpredictable and dependent on sort of the past evolutionary contingency of the pathogen. And it's true, like if you think about hospital-acquired bacteria, they've been exposed to selection by many different things already. So it's completely different game than just taking your bacteria and see what happens with that.
This has been super cool. Anybody else have more questions? There you go. Come up. Wait. There you go. Hi, Joseph.
Okay, how are you?
You have a question?
Goes to yes, one for John Paul and Ville. Are there instances when the bacteria is able to develop this resistance to both phage and antibiotic?
You mean able to be resistant to both?
Yes, antibiotic and phages.
Yeah, we've seen experiments where we expose them to, like, we get positive correlation between the phage resistance and resistance to aminoglycosides. And I think, like, we haven't tested this explicitly, but just looking at colon morphotypes and their resistance, the resistant ones were quite mucoid. So it could be something to do with the diffusion of antibiotics and blocking the receptors. Yeah. And I think there's lots of examples where people find this sort of positive correlation between these two traits. Yeah.
Okay. Yeah, what about John Paul?
Yeah, I guess probably this happens in vivo also, but I don't think you will see them. When your bacteria becomes resistant to the antibiotics you are using and also to the phages, it's just a failure of your therapy.
Yeah.
So I don't think you will necessarily see them. In vitro, you can in certain experiments you can of course select them and see them, but I don't think it will be easy to do in vivo.
Okay, awesome. Thank you.
No worries.
Anybody else have questions? It seems that nobody else has more questions.
I have one more question. So I was wondering, I was reading your paper from 2015, and I was looking at the graph of the chronic bacteria versus the intermittent bacteria, and how they seem to be more susceptible to this evolved phage. And I was wondering if you could elaborate on those findings. Sorry.
So, no worries. Because that was a kind of interesting pattern, and then this isolates was kind of just chosen a bit random. So unfortunately, we didn't have sequences of them, so we couldn't kind of compare, for example, if there's any similarities between the ones we chose. But they did show like how long they had spent in the lung had a clear effect. And maybe it's, I mean, like, we haven't sequenced those clones, we haven't really done any work with them afterwards. So not 100% sure what's going on there, but we're working on sort of better established phage bacteria collection now, so maybe we can see some of these similar patterns depending on what sort of infection states they are isolated from. Sometimes with this data, the problem is that with Pseudomonas and CF isolates, you see so much variation and variance, so it's hard to kind of pick up the mutations that might be causing this unless you kind of replicate those in some sort of ancestral background. So it is very challenging to determine the genetic determinants still.
Yeah, I just thought it was interesting because we deal with patients with chronic infections, and we're those are usually the cases we have to take because they're stable. So I'm wondering if phage therapy or these evolved phages would be a better option for these types of patients.
Yeah, potentially, and just using the clinical isolates to train the phages in the first place, try to kind of see if you can make any of the phages we have in the lab better infecting them. And I think in this sense, like when we try to tailor when we have the infection and we want to have a phage that would be able to do the job, I think training would be a potential answer. At least with Pseudomonas, it seems to be doesn't take too long. You can kind of get trained phages, at least with the pure one, in less than a week. With Ralstonia, it tend to take longer. We were kind of starting to see effects after 2 weeks. Before that, we didn't see much happening. So it's like we're looking like, okay, I guess nothing's happening in here, but we still kind of maintained the experiment, and then we started to see a nice increase in inhibition.
But it's dependent, so appearance of evolution in phages is dependent on the bacterial replication cycle, on how long that takes. So for example, a bacteria that grows slower like Ralstonia versus a bacteria that grows faster, just like Pseudomonas, is there a difference there?
Probably. Even with the same bacterial strains, like if we manipulate nutrient concentrations, the phages that are super effective at higher nutrient concentrations don't seem to have much effect when we use very low nutrient concentration. So if this is due to replication, or they're not expressing the receptors at low nutrient concentrations, I'm not 100% sure yet. But like changing the environments, you definitely changed the infectivity of the phage as well. So more variable environments you're working with, more variable your outcomes will be as well.
James.
Hey, how's it going again? I think actually my phone cut out a second. I wasn't sure if it was our same question, but one of Billy's papers, I'm trying to remember which one now, showed that the co-evolved bacteria was actually more susceptible to an earlier point phage. Is that right? Got that right round?
I'm not 100% sure, to be honest. Which paper you're talking about, but possibly, yeah.
It was yours and was it you and Bucklin or Brockhurst? Let me just pull up my note here. It was you and Bucklin, so it was this idea. It was a co-evolution paper. It was your 2013.
Oh yeah.
So I mean, that paper made me feel so much better about my life because we found the same result, and I was like, how is this? Like, what happened? So to reiterate, we did a co-evolution experiment. We found our endpoint bacteria were actually were very susceptible to the original phage. So we've gone through this like co-evolution experiment and then added back in repeated the therapeutic and found it was still sensitive to that. The point about mechanism here, we think it's a combination between two targets of the phage overlapping with the antibiotic target as well. So this is using Paul Turner's OMK1 phage. The idea being some gyrase and efflux are both targets of the phage and the antibiotic.
Yeah, I mean, like you don't know. The experiments don't always go the direction you would expect them to go, and you do get these sort of weird results. And I think it's important to publish them as well, because probably like as you said, when you get a weird result, you're probably not going to be alone with that. So other people might have found the same thing, and but if no one publishes, then it never comes to light, and people don't discuss about that.
Right. So I saw that in the paper. I was like, oh, thank God! I haven't screwed everything up. Someone else has seen the same result, and it's really I trust his work. So I was like, okay, cool. I'm happy to cite your work and say you guys found it.
Yeah, I mean, like with phages as well, like you do get like quite a weird. Sometimes like between replicate variation.
Yeah.
Sometimes you don't. Is it because of you have bit different MOIs or what's going on. So you know, I mean, like I find the phage work can be quite temperamental at times as well, and then you find this like, why is he doing now that? And like, what's going on? And then you kind of keep repeating, and then you do see some very weird stuff. So what we've seen, for example, one in the temperature experiment, we have a phage that doesn't have effect on the bacteria. When we increase the temperature to 34, the bacteria starts growing much better in the presence of phage. And you know, feels like, oh, there must be an error. We've done that now 3 times and it happens. So, but no idea what was going on there. But you know, it's a pattern and consistent, and we can reproduce that. And then, yeah, who knows?
Yeah. Oh, we'll keep going, right?
Yeah, cool.
I'm going to drop off, but I hope to see some people at Evergreen in a couple of weeks.
Yeah. Jean-Paul, you were going to say something.
Oh, yeah, just I realized that I gave a partially wrong answer to Michael earlier. So, yeah, of course, we see those bacteria that are resistant to phages and antibiotics, and we do isolate them. You do see them. Sorry for that.
You're good. I hope to see everybody at Evergreen. Tom, you came up. You have a question?
Yes, hi. Oh, wow. I'm really lucky then. Been quite interested in this topic. And so my general question is that how much is characterized about the bacterial and archaeal phages in the gut? And do you have a good idea of the survey of them where what percentage of them are obligatory lytic and how many of them are obligatory lysogenic? And I have a few questions beyond that, but I think this is the most important question. Thank you.
Thank you. It's an excellent question, and I can just answer straight off. Like I don't have any idea, but like we've been starting to look a bit of the archaea in the rhizosphere, and then we've been starting to look phages internal in the rhizosphere, which is not the gut, but it's kind of going beyond your sort of pathogen and its associated phage. For any doubt, probably they are playing an important role. And you know, but I don't have any idea. I wouldn't be able to answer this question. But I would expect that people will you have lots of metagenomic sequencing studies going on, so we'll get probably more information about that. And for example, at the York University, people are working a lot of with wastewater, like anaerobic digestion systems, and using long read sequencing to try to kind of understand a bit of the metagenomics and try to kind of construct some larger genomes as well. And I'm also interested in looking at phages in this system as well. So maybe that will also give some insight, like if they are there and how important they might be. With the soil system, we have the same question as well. Like, okay, are these phages we can kind of identify from a metagenome set? Are they lytic or lysogenic? And I think it is a difficult question to answer still. And I think we just need to be characterizing more stuff and improve our sort of databases. So and then do more experiments in the lab to test what's going on, I think.
Thank you. That's yes, because many of the microbial interactions in the gut is syntrophic. In other words, one microbe cannot degrade a particular compound unless they have the help of the other hydrogen scavenger. Yeah. And one thing that I'm wondering is that if you have a targeted destruction of that one partner, I don't know, but I'm thinking what are the downstream effects of that. But I do like bacteriophage as a way to target organisms.
Yeah, I think that's a really good idea, and then I think that's something to test for sure. And I think that's also like I like the idea of like going beyond just like, okay, we have a pathogen we want to kind of remove. You could also cause all sort of like indirect effect in communities by if you have dominant bacteria, maybe you can introduce a phage that is targeting that, and that will kind of create community reassembly and then change the functioning of community potentially into a better direction. So I think instead of like adding things into community, you would probably steer the community more by just like removing some important links, as you say. So I think that's a really good idea and interesting idea.
Thank you, Ville. Now, as the last question, and because it's Friday afternoon / night where you are, I have a question. Do you have your guitar by? Right close to you?
I don't have guitar close to me, no, at the moment.
Oh man! So we're missing your all the musician career that you have. We missed the tune, the Phage Friday tune.
Phage Friday tune. I'll come up with something on my computer, and I'll send it for the next time or so.
Thank you so much. Thank you so much. Well, we hope to hear it then.
Cool. Thank you.
Jean-Paul, you're gonna.
Yeah, I think I have a question. So a lot of persons in this group might wonder, how do you do this? You're a productive researcher and you're also a rock star. How do you do this?
Well, I don't. So, I mean, like, kind of, I'm not doing that much touring at the moment because of, it just can't be at two places at one time. So, there was a time a few years back when I was just like writing crowns at the backstage after the show, and then try to kind of multitask, and it just didn't work. I'm still in the band and I'm working on the music, but I kind of left behind most of the live shows now because it's quite busy. Well, of course now it's been really super quiet, but normally there's quite a lot of tours all the time, so it's impossible to be in two places at once. But yeah, I mean, like, the band's been going for a long time since we started when I was 17. So it's been kind of part of my life, well, as long as I remember. So I think the science have kind of crept in the middle of that. But yeah, I mean, like, I still like to keep keep myself busy, composing music at home, playing guitar. But also it's like I really like science as well, and if you have a research group and then teaching and whatnot from the university, it's just like too much.
There's cool people in this world.
Well, I don't know about that.
That is super cool, though.
Yeah, like, I think it's amazing.
I was in a choir. That doesn't count.
Yeah, being backstage and writing grants just sounds like a challenge.
Yeah, I mean, like, it's like hell on earth when everybody else is drinking and I'm staying up and yelling, and then you're kind of like, okay, I would like to have some sleep. But I had really like loads of like, I remember this US tour, maybe 2015. I wake up fresh 9 a.m. every morning, and I had like 5 or 4 hours to just write grants and then do my stuff. Everybody else was sleeping, and I'm hangover.
Wow. Okay. That sounds difficult. The life of a rock star and the life of a scientist don't seem to be very compatible.
No, they aren't.
Ville, thank you so much. Thank you so much for joining us in Phage Friday. Josh, it was nice to see you again. Thank you, everybody in the background and down in the audience. Please feel free to ask questions. Also text us. We're on Twitter, on Instagram, and we're also here on Phage Club every Friday. Next Friday, we're going to talk about Phage satellites. I don't have the time yet. I think it's going to be still 1 Central, but I will let you know. We have somebody invited who's going to talk to us about Phage satellites and what that means. Oh, and also, We have Phage Wednesday this Wednesday, and we're going to talk about Phage Australia. So we have two of the directors of Phage Australia coming over and talking to us about Phage Australia, and it's going to be on Wednesday, 9 p.m. Central. We hope to see you there for those who can join, and we hope to also record it so people that cannot join will be able to listen in.
It's on Clubhouse?
I think that's... Yep,
okay.
Clubhouse.
Okay.
Yeah, we're going to do it on Clubhouse. We were trying to think... We were thinking of switching to Green Room maybe, but I think Clubhouse, most of the people are in Clubhouse now, so there's that.
Okay.
It is awesome, people, to see you. I hope to see you all in Evergreen. Evergreen's coming up in a week. Yeah, well, yeah, kind of in a week and a couple of days. I hope to see you all at Evergreen, and have a great Phage Friday.
Bye, everyone.
Cool.